To introduce Wen-Chieh and McKeith Pearson, both grad students in the Department of Biological Sciences. And I believe Claudio Aguilar is your advisor, is that correct?
Yes, that's correct.
All right, very good. Longtime participant of our center. Very good. So they're going to talk to us today about the development of algorithms for the automated quantification of biological imaging data. So if you have any questions or comments, please enter them into the chat window and we will get the presenter's attention. Otherwise, we will let them take it away.
Okay, how now we are going to start. Good afternoon, everyone. My name is Wen-Chieh.
My name is McKeith Pearson. We're from the Aguilar Lab.
Hi, so today we are going to talk about development of algorithm for automate quantification of biological data. So as biologists, we always try to understand the nature better. One of the way of doing it is to look at the difference between normal condition and abnormal condition. But for example, for morphological differences or distribution difference of certain proteins or expression levels difference between the conditions. And some of the way of doing it is to take pictures of the sample and look at the difference between the sample from different conditions. So that means, with that being said, that means we need to deal with a lot of image data analysis, which would take time, and also it would introduce human errors to the data, so that will lead to may cause some problem for the results. So here we want to introduce algorithm to automatically quantify the data for us to facilitate the step and also decrease the human introduced errors. So I'm going to start with my part with one signaling pathway that I'm focusing on, which is notch signaling pathway. So notch signaling pathway is a very important signaling pathway during embryonic development, especially for neuron development. Also, in adult cell, it involved in cell proliferation, angiogenesis, and invasion. And by its name, you know if they are misregulated, they will cause cancer. So let me briefly introduce how notch signaling pathway works. It involves by two adjacent cells. One is signal sending cells here, and the other one is signal receiving cells. So on the signal receiving cells, there is a protein called notch on the cell surface. And on the signal sending cells, there is a protein called delta, which is a ligand of notch proteins, meaning that they bind together. Upon the right moment, something wrong with the show. And in the protein called Epsin will recognize delta and internalize it into the signal sending cell through a process called endocytosis. You see, since delta and notch are bind to each other, this movement of delta will cause the stretch of notch protein on the signal receiving cell, which will cause the initiation of the signal in the signal receiving cell. With that being said, this internalization step is the triggering of this notch signaling pathway. In a lot of cancer conditions, they are this notch signaling pathway are upregulated, meaning that this internalization of delta could be a druggable target. So how do we attack this question? So we use yeast as a model system. Since the Epsin recognized its cargo, this mechanism is highly conserved in across the evolution. in yeast, in body yeast, they also the yeast Epsin also have a cargo called ENA1 that's specifically being internalized by Epsin. And since yeast are unicell organism, it's largely simplified the experiment condition that we have, so that will help a lot for us to do the study. So the method we do it is that we take ENA1 protein with green fluorescence protein so that we could track the distribution of the protein. As you can see here, that this cell, we have some ENA1 protein on the cell surface, also in the inside the cell. And also, these are two common phenotype of the yeast cells. One is like oval shape, and the other one is budding. So you can see two cells, two circles that are connected together. So we use yeast as a model system to design a drug screening system. So the way we do it is that we express ENA1 GFP and turn off the protein production and let it go for 5 hours. Within these 5 hours, ENA1 GFP will internalize into the cell and eventually be degraded by the cell machinery so that the cell will become dimmer compared to the initial time point. So the brightness of the cell can be distinguished by an instrument called flow cytometry, a method called flow cytometry. It could sort out the bright cells and the dim cells. So within these 5 hours, this is the right window for us to do the drug screening. So we use this low-pack library, which are from library of pharmacological active compound. Most of the compounds in this library are FDA approved or they are derivatives, and there are about 1,300 compounds in this library. Within this 1,300 compounds, we expect to some of the compound does not do much, so the result will be vehicle-like. Some of them will enhance internalization and degradation, so the cell will be dimmer than the vehicle-like cell. And some of the cells that will be brighter, which are internalization inhibitors or degradation inhibitors. So these cells will also be brighter than the vehicle-like cells. So here is the question. So now from the flow cytometry, we can only know these cells, these two types of cells are brighter than the vehicle-like cell, but distribution is unknown yet. So we need to use a microscopy to examine the distribution of the proteins. So in this case, this drug is inhibiting internalization, so you can see majority of the protein are on the cell surface, not too much in the cell. And this one, most of the protein are internalized, so not much on the surface, but mostly inside. And this one are intermediate. So how do we rank these candidate compounds? We know by eye that this one will be our best option, and this one will be the least option. But however, there's so much in between. How do we rank them? So we need to have a quantification method, which we have here is to quantify the fraction of intracellular fluorescence. What that means is that all the fluorescence intake in the cell, what's the fraction of it inside of the cell? How do we quantify it? Is that we use this equation here. Outlining the cell, we can get a total fluorescence intensity of the whole cell. And outlining the inner part of the cell, which is from here, you see we can record the internal intensity of the cell, and then we subtract with the background so that we can get this result. However, as you can see that I'm manually tracking the periphery of the cell, this step will cause human-introduced error, and also it's taking a lot of time. So we are aiming to develop a automatic fluorescence distribution quantification algorithm together with Wen-Chieh Liu. We are the CSOI team research funding awardees. Starting from 2015, we started develop this algorithm. So here I'm giving you a example of the image that we are dealing with. For example, in this image, it has a couple of cells. Some of them are bright, some of them are dim. So we cannot apply a single background to all of them because the brightness of the cells are different. So how are we going to attack the problem? So this is the structure of the algorithm. From the original image, we are going to separate the cell into components, and then we are going to define the component boundaries, and then get the total intensity of the cell. Following that, we are going to define the internal boundary of the cell and take the internal intensity of the cell. Then we are going to define the internal background, which is this, and get the internal background intensity. Then we calculate the ratio. In this 5 steps, there are some sub-steps which are listed in here, but I'm not going to go into detail on this slide, but I'll go through the whole process in the following presentation. So far, does anyone have questions? Okay, I'll continue. So the first step is to eliminate the global background from the image. We use different steps of threshold to eliminate the global background. We apply 5,000 thresholds to this image. For each of the threshold step is the maximum pixel intensity, is the difference of the maximum pixel intensity minus the minimum pixel intensity and divide by 5,000. As you can see, once we apply the thresholds, the background is going to be eliminated, and at the later thresholds, you also eliminate the cells. So we need to find a good threshold that only eliminates the global background but not the cells. So in order to do that, we binary the image from each threshold, and this is how it looks like. In each of the threshold image, we could cluster the adjacent pixels into components and make a graph like this. So the y-axis is the number of components, then the x-axis is the threshold number. Now, as you may see here, we only take even though we have 5,000 threshold, we are only looking for the first 500 steps, because we know that from the test we did, we found that within the 500 steps, we could find the good cutoff. And here is an assumption here. So when I take the image, I try to keep the cells apart, and I only take from 6 to 15 cells per image. So potentially these beings are my targets. So, and here are how the image is looking like. So the good cutoff we choose is that a cutoff where the number of components persists for a couple of times, so that we know that the size and the position of the component wouldn't change for a long time, meaning that that's a higher probability that those are the cells that actually locates. That's the position where the cells are actually locating. So here, since this string of threshold persists for a long time, so we choose this. The algorithm would choose this image as a cutoff image, and from here we could separate the single components based on the coordinates of these components, and we separate it into single components. And I'm using this cell. I'm using this component as an example for the following steps. So now we have this black and white image, and this represents the location of the cell. But how are we going to get the real pixel value from this image? So what we do is that considering this image as a matrix. So everything inside of the cell, we give it a value of 1. Everything outside of the cell is 0. So you have a matrix looking like this. And from the original image, it's looking like this. It has its own matrix, and each of the pixel has its own true value of the pixel intensity. So if we multiply this metric with this metric, we could get something like this, meaning that everything within this area will have the actual pixel value, but not the background, so that we could isolate this region from the original image. And from here, we get a cell. However, as you may see that here we have local background that is not properly define the cell boundary. So the second step we have is to define the cell boundary by eliminating local background. Using similar idea, we apply a series of thresholds that will filter out the pixel intensity from low pixel intensity to high pixel intensity. The assumption here is that through the process, since the background pixel intensity will be similar, so we will be eliminating the backgrounds at one particular threshold, and that is the inflection point on the curve. So we found that the second inflection point often to be the right cutoff to find the boundary of the cell. So we choose the second inflection point as our cutoff for finding the boundary of the cell. Okay, so now we found the boundary of the cell. As you can see that the background has been eliminated. However, also during the process, we also eliminated some of the pixel within the cell, or even make holes on the cell boundaries, because the pixel intensity is similar to the background. So how do we, so if we take the total intensity from this image, we will be underestimating the total intensity. So how do we solve the problem? So we need to fill these gaps. So here I have a representative image here. So if we zoom in this white box here, red box here, so this means that how it looks like the pixel by pixel. In the gap, we have the black pixels, and here we have the white pixels. And we give the values to each of the pixels, the black pixels, numbering here. The number means how many pixels in this 3 by 3 square are white. For example, in this one, there are only 3 white pixels, so the number is 3. In this one, there's only 1 pixel in this 3 by 3 box is white, so the value of it is 1. So we could ask the algorithm to fill up any pixel value above 2 to white, so that we will get something like this. So as you can see, these 2 and 3s will be filled up. So we seal the gap here. And the next step is to fill in the holes in between, so that we ask the algorithm from vertical, each vertical columns, if there are two white pixels that is covering the ceiling and the floor, we're gonna fill in with the white we're going to set them into white pixels, and same for the horizontal direction, so that we could fill all the gaps. And from here to here, we have a whole complete cell filled with white pixel. And again, we could mask back to the original image and get a cell looking like this. So here is, we can use this filled image as our complete cells. So we want to use this as our total intensity. However, if you look close to the cells, you will find that in this mother cells and the daughter cells, the local background of the mother cell is slightly brighter than the daughter cells. So if we are using the whole cell to find the local background, then we might not be accurate. So in order to do that, we are going to in order to get the more accurate total intensity, we are going to separate the mother cell from the daughter cell. The method we're doing it is to rotate this cell horizontally and split it into two, and ask the algorithm to find the trough of this hemicells on this side and on the other side. And then we joined these two dots, and then we could get two separate cells, one daughter cells and one mother cells, and we see them as two separate cells from now. And of course, there are some cells that doesn't have the budding parts, so this step wouldn't cut this kind of cell. So from here, we code these three cells. We set these three cells as single cells and take the total intensity of them and fulfilling the equation. And the second step is to define the internal intensity of the single cells. With that being said, what we want to do is to eliminate the cell membrane, so turning this cell into this kind of cell. The way we do it is to shrink the pixel towards to the centrioid of the cell for certain pixels, because the thickness of the cell wall or the cell membrane are homogeneous through the cell. So we could do that to shrinking few pixels and get a smaller version of this one and get the cell shrink, and then we can go the internal intensity. So far we are still optimizing this step, because you can see most of the cells they are looking good, but for some brighter cells, even if we shrink the membrane, we still have some leftover here, and that would cause some inaccuracy for the background detection. But we have a pretty good idea how to solve this problem. So let's continue with this. So after this, we want to determine the internal background of the cells, meaning these guys. So the way we did it was to make a histogram based on the pixel intensity and the probability of the pixel intensity. And since these guys, the backgrounds are dimmer, so they are located in the first peak of this graph, meaning that this peak is the background, this peak is the background, and here is the shoulder, so here is the peak of the background. So by eliminating the background, we can get the total internal part like this. And within the previous the value from the previous step, we could complete the whole equation and get the number from each of the cell and get the distribution representative, the true distribution, present distribution for each of the cell. So based on these values, we could rank the efficacy of the drugs. And this is how my algorithm was designed, and we are still optimizing some of the steps, but we have a pretty good idea how to do it. And so far, does anyone have questions? If not, I will let McKeith take over.
So good afternoon, everybody. Again, my name is McKeith Pearson from the Aguilar Lab. I am a third-year graduate student. So I'm going to be talking to you about my portion of the project, where we're developing quantitative methods to analyze cell morphology. So cell morphology, or cell shape, is an important property that biologists exploit to be able to quantify different aspects of cell types. Take, for instance, cancer. Particularly in the bottom rows, you'll see that the morphology, or the cell shape from normal versus cancer is strikingly different. So biologists, as you may know, take use microscopy heavily, especially cell biologists when they're looking at images, and they analyze most of these images either with image analysis tools or by hand. And so some of the analysis by hands can be due to errors, when Jay was saying. So an important aspect of this project is to develop an algorithm to analyze cell morphology. And then a broader impact of this project is to start to eliminate or take away the subjective analysis that some biologists still use for quantifying data that goes into journals. So again, we'll talk about Epsin. So my project has to do with Epsin and a protein known as Huntington interacting protein 1, or HIP1. So Epsins are extremely important in establishing polarity in morphology. Actually, it was discovered in our lab that expression of Epsin 3 in particular leads to morphological changes in epithelial cell. Particularly, epithelial cells expressing Epsin 3 acquire a more migratory morphology and migratory properties. This migratory properties is very indicative of epithelial to mesenchymal transition, which is it could be a death sentence in cancer patients. So what I mean by polarity is that if you notice that the proteins are not being localized evenly around the cell, proteins are actually into a particular region of the cell. So when you think about proteins doing specific functions, if this protein's job is for cell polarity, or morphology, is important that it's on a particular side of the cell, and that will determine its overall morphology. So introducing Huntington interacting protein 1, HIP1, shares multiple functions with Epsins. In fact, HIP1 and Epsins have been both previously known to be important for endocytosis, as well as cell polarity establishment and morphology. In fact, Epsins and HIP1 are known to actually have interact not just in mammals, but across multiple eukaryotic organisms across evolution. But the precise mechanism in which they interact is actually not known. So since Epsins and HIP1 are evolutionarily conserved across multiple organisms, we decided to try to answer this question using yeast. Since yeast are much simpler organisms, and we can manipulate them genetically with more ease than we can with mammalian cells. So there happen to exist several homologs or different versions of the same protein. So yeast have what is known as Epsin 2, and what I'll be calling yeast HIP1. So if you express these two proteins full length in a cell, you would expect to have normal cell division. And here's what normal cell division for yeast look like. You have two instances. One where you just have a circular cell. Yeast have a circular typical morphology. And then you have what looks like two circles fused together. That is a cell undergoing cell division. And so the type of morphology we want to look at that Epsin 2 yeast HIP1 brings on arises due to cell division and sometimes cell division defects. So when we overexpressed Epsin lacking a portion of its full-length protein, then we also observe not just normal cell division, but also aberrant cell division defects. So when you're talking about morphology again, obviously this cell type has different morphology. It has several heads as opposed to the typical one or known. And so we decided to go ahead and analyze basically multiple regions on Epsin and see if we're able to discover a cell division defect. And we didn't just knock out regions on Epsin, we knocked out regions on yeast HIP1. And so we were hypothesizing that if we see cell division defect, then that region that we just knocked out could be important for interacting with the interaction of Epsin to HIP1 for the normal establishment of cell polarity. So let's talk a little bit more about these morphological defects that are caused by Epsin and yeast HIP1. So you have, first of all, let's just compare. This is what you'll normally have. And normal yeast cells have an average of 1 nuclei per cell. The first morphological defect is extra cell wall. So yeast normally have little to no basically opaque regions. What you see here would be a massive deposition of cell wall, and that's considered a defect. This is not typically found. What's also not typically found is 2 budding events. It's also another defect. It's known as type 2. Then you have increasing severity. So instead of 1 budding event, you have a single premature budding event before this cell has split off. That's known as type 3. And then type 4 is the more typical severe cell division defect with multiple budding events. So the methodology is, again, to use combinatorial analysis of either full-length Epsin or HIP1 or their different truncations to test for relative severity of cell division defects. So what we did was, once we expressed these proteins in yeast cell, we visualized them using methylene blue stain microscopy. The methylene blue is the dark dye that seeps into dead cells, and it allows us to distinguish the cytosol from the cell wall with ease. Next, we are to basically quantify the amount of normal versus type 2, type 3, type 4 in the image. And so let me break down basically what that means if I were to do this subjectively or by hand. So these experiments require 36 possible combinations. And for statistical significance, these experiments need to be repeated 9 times. So that's 36 times 9, which is 324 experiments. And then that's just, so each experiment will require 9 images. So that's also 9 times 324. So it gives you around 3,000 images to analyze for one project. So it would be a good idea to have an algorithm that will be consistently analyzing these cell division morphologies, and so also to ease some of the work that the graduate student has to do from counting them. So let's go to the algorithm. So how does the algorithm work? So I'll be explaining the image processing part towards the end, but you would have an image, and then you'd break it into its binary compositions. You would convert the image to a binary image. Then you would analyze each of the cells individually. So the first test that they would be put through was something called circle test. And it utilizes the properties of the area inside the cell versus the area of the circumscribed circle around the cell. So the algorithm draws a circle around the cell, and then it takes the ratio, the area inside in pixels, and the area of the circle in pixels, and develops a ratio. Our thinking behind this analysis is that the same test put up to a cell with an abnormal cell division defect or an abnormal morphology will have a lesser or a lower area ratio, so the ratio of the cell over the circle. And so that's the cutoff that we are hoping to generate. And so we have generated a pretty clean cutoff. So we analyzed about 40 cells, and we basically wanted to know what was their area ratio. So to the y-axis is each individual cell's area to ratio, and then on the x is the type of cell. So these are normals, type 3, and type 4 here. And as you can see, there's a nice cutoff of about 0.48. So 0.48, if we were to take these cells and run it through the circle test, if you were analyzing the ratio of the area of the cell to the circle, you would have a cutoff of 0.48. Anything above 0.48 would be considered normal in type 1 category. Anything below 0.48 will be type 3 and type 4. So now that we have a good separation of normal, type 1, type 2, and type 3 and type 4, let's focus more on how we're going to individually analyze this first box, which is normal, type 1, and type 2. And we'll leave type 3 and type 4 for later. So we're going to subject normal, type 1, and type 2 morphology cells to what is called radial analysis. So radial analysis takes the cell and finds the centroid of the object, and then draws an arrow, looks at the length from the centroid to the last pixel in a radial motion. That's why it's called radial analysis. So this generates basically a graph of certain lengths from the center to the edge of the cell, and that's plotted here. So from 0 to 360 degrees, you have these different lengths that are represented by the arrows. As you can see, for this type of normal morphology, you don't have any where we see local maximums. It looks like a flat line. And this other type of normal morphology, you actually do have 2 local maxima. And so the local maximum will, any local maxima that is 2 or below will be considered normal. So how are we differentiating type 2? Well, type 2, if you subject it to the same test, type 2 demonstrates not 2 local maxima, but 3 local maxima. So this is a good way to separate type 2 from normal and type 1. So next, now that we have a separation of normal from type 2, we're going to focus on how we're going to separate type 3 and type 4. And we're going to do that with a circular ratio test. So what this test does is it finds the circumscribed circle, the circle that fits exactly around the object, and then it's going to find the inscribed circle. So that's the maximum circle, the biggest circle that can be inscribed in this object. And you do that same analysis, and then you want to take the ratio of the circumscribed circle divided by the maximum inscribed circle. And that develops a nice cutoff of about 8 when you're trying to differentiate type 3 and type 4 cells. So when you subject your type 3 and type 4 cells to the circular ratio test, you have a cutoff of about 8, and then you'll be able to separate your cells from type 3 and type 4. Also, I want to note that the cell wall test still needs a lot of work on it, and we will in the future discuss more methods that we can think of that will separate the cell wall from normal. So in conclusion, just to remind, Epsin and HIP1 are evolutionarily conserved, and they are speculated to be involved in regulation of cell morphology. So we have developed an algorithm, or still developing an algorithm that will quantify cell division morphologies in budding yeast, and with the expectations that this data will allow us to understand which regions of Epsin and HIP1 are important for regulating the cell morphology. And also, this type of research is good because it is trying to, again, get away from the subjective analysis and have computers more objectively and consistently analyze our data so that our data will not have high variability from either from lab person to lab person or from different labs to different lab. So with that, I'll go to my future work. So again, I would need to develop, again, other methods for working on a cell wall test. And since a lot of these programs, a lot of the things that I want to do, like convert my cells to binary and extract individual cells, can be done in Fiji, I need to understand Java as well. And particularly Fiji offers pretty good methods for separating your cells. You have a raw image such as this, and Fiji has an already application for converting to binary, and also particle analysis. It is able to identify individual cells. So it's something that was recently found. So it's something that we want to implement into the algorithm that we have. And then finally, we have a function that will then split up the different objects so we can analyze the cells individually. So with that, I will conclude my presentation. I'd like to thank some people. So Felix Francisco Sanchez, Michael Young, they were the original people along with me that helped me on my project. And then the Center of Science for Information, of course, for allowing giving us a platform to discuss our scientific data, and for the funding as well. And obviously, National Science Foundation. And then particularly Dr. Claudia Aguilar, our PI, and the Aguilar Lab. and also Purdue University Department of Biological Sciences. So if there are no further questions, I'd like to thank you.